So in this lesson, we're going to take a look at the variation that exists within a species due to two processes that deal with sexual reproduction: meiosis and fertilization. So within every single population, whether you're talking about butterflies or lions or even cacti, there exist different variations within color. As you can see in the wings of each of these butterflies, there's different shapes and different patterns. As when you're looking at the variation within lions, all sorts of different colors of manes, patterns, shapes and sizes, and even within the plant kingdom, you have cacti that are different sizes and different shapes, different colors, different patterns. So how do we get all this variation within the same gene pool? So again, the two mechanisms for creating variation within the populations are meiosis, which is the cell division process that you studied before, taking one cell and dividing it up into 4 gametes, as you can see here, whether they're sperm and egg, in order to pass those on to the offspring. And the second mechanism, which is called fertilization, the act of taking the DNA in a sperm cell and combining it with the DNA inside of an egg cell, and getting a new combination of alleles. So just a quick overview of meiosis, just to revisit what we learned before. We start with a cell that has a diploid number of chromosomes, 2n, and we end up with 4 individual gametes, which are each going to have a single set of chromosomes. We call that n, haploid cells. So the whole process of meiosis, the point of it is to reduce the number of chromosomes. But what you'll notice here is that somewhere along the way, you end up with chromosomes that have recombined their genes. So they're not completely all red, and they're not completely all blue. And they're a combination, a recombination of what the original ones were from their mom and dad. So the part of meiosis that we're going to concentrate on is here in the middle. We're going to take these 2 homologous chromosomes, which you can see are the red and blue, which I'm using slightly different colors down to the bottom. We'll just say that the red chromosome is from the female here, and the green chromosome is from the male. But they contain the same genes, the same traits, so they're homologous, they're same size, same length, same shape, same location of their centromeres, as we can see here in the middle. They just may carry different variations of the genes. We call those alleles. So at some point in meiosis, we observe that these 2 chromosomes, one from mom, one from dad, they cross over, they recombine their genes. So they're basically trading alleles back and forth at random rates, and this actually happens in a process called prophase 1. That's the step that you studied when you were looking at the process of meiosis before. So after the cell has completely gone through the stage of prophase 1, then we end up with both homologous chromosomes having traded genes. Both now carry a recombined sequence of DNA, a new combination of alleles that is very unique to this particular cell. So as you can see here on the left, we have these unique bands that represent the alleles, and those alleles have exchanged. So you have a recombined DNA on all 4 of those sister chromatids. So at the end of this process, every single one of the gametes that's produced is going to have a very unique combination. There's 1, 2, 3, 4 sister chromatids here that each are going to be separated into the 4 different gametes. So every single one of those gametes created by the process of meiosis is very unique and has their own separate combinations of alleles. So that's the first mechanism that we use to explain why there's all the variation within a population. So following the process of meiosis, and we've now created your sperm from dad here with all the DNA here, and the egg from mom, which has half the chromosomes, haploid cell with her set of recombined alleles. Then we can combine those together to get a zygote, a single-celled organism. And that gives us here, the zygote has a combination of all mom's genes, all dad's genes, and we can take those genes out, and you get all this DNA here that's all mixed up. So instead, we're going to take it and we're going to organize it into a fashion by size. And we call this picture of all the chromosomes inside of a single cell a karyotype. So this karyotype really shows us the variation that this organism has. It's very, very unique to this organism only because it's a combination of the sperm and the egg that was created through the process of meiosis. So again, from the process of meiosis, we created gametes, which gave us 23 chromosomes, one from each of these pairs here, and 23 chromosomes, one from each of these pairs from mom. And when we combine those together, you end up with a new combination for the kid. That new combination for the kid really has one chromosome number 1 from dad and one chromosome number 1 from mom. And because they come from separate parents, then the chromosomes are very unique to that particular individual. And because each of these offspring come from separate parents and separate sets of genes, then you end up with a new combination for every single one of these offspring. And so that explains why we have such a huge variation within a single population. So together, these two mechanisms of meiosis and the process of fertilization, we can get just an infinite amount of variation within a population.